Hello, Assalamu alaikum. The Sheikh Hamdan bin Rashid Al Maktoum Award for Medical Sciences, established in 1999, has been a driving force to support the medical sector. It collaborated for decades with academia and the healthcare sector to fund distinctive research and honor excellence in service. These efforts have paid off well, as two Grand Hamdan Award winners were awarded the Nobel Prize in Physiology in 2010 and 2020, Sir Robert Edwards and Dr. Harvey Alter. Education was one of the leading awards' objectives. It aimed to provide opportunities for capacity building activities. Hence, this new series of awards winners webinar will cover various infectious disease topics. Before introducing our esteemed speaker today, I would like to remind you that the question and answer window will be open, and I encourage you to write your questions earlier so that we can filter them and provide them for our speaker. All the attendees also will receive an email to download the attendance certificate within the coming days. Here I would like to welcome Professor Marc Laforce, winner of the Hamdan Award for the Medical Research Excellence for Prevention. Professor Laforce is the Director of the Technical Services at Serum Institute of India Limited and the Clinical Professor at New York University, Langone School of Medicine, New York, and was the Director of the Meningitis Vaccine Project till 2012. Professor Laforce, the floor is yours.
Thank you. It's an honor to participate in today's session, and I am grateful for the recognition of the work that an entire team has done over the last 20 years, and I find it humbling to represent them. But I would like to do so and to, at the same time, tell you a story. And that's a story of the development and introduction of a new meningitis vaccine in Sub-Saharan Africa. I am going to attempt to do my share screen. Here we are. Is that visible to everyone? There, thank you. This is a description of the problem as it was in, I would say, 2001, and represented a problem that was existent for 100 years. This huge area in Africa called the Sub-Saharan Africa contained during that century the nidus for about 90% of the world's episodes of bacterial meningitis. And one strain accounted for all of that, and that strain was Group A. And this is a description of the disease burden from 1948 to 2013 or 2014. And I call your attention to the ordinate, that is the number of annual cases. You see these huge epidemics that were codified by the World Health Organization beginning in 1950 with these tremendous peaks about every 10 years. And they culminated in 1996 with an epidemic of reported cases at 188,000. these are just enormous numbers, and the amount of chaos that was generated during these particular epidemics is briefly summarized in this slide. There's no way of predicting where epidemics occur, and so it's not where hospitals are, it's where people are. And so what we were able to see as epidemics occur were these awful examples of not only disease, but of the chaos that was rendered during these particular epidemics. The situation in 2001, at the time when we examined this issue seriously at WHO and with the Gates Foundation, revealed that at that time there were only these polysaccharide vaccines that were available, and these were largely used in reactive campaigns. By reactive campaigns, I mean that once epidemics occurred, governments felt obliged to do something, so they would obtain polysaccharide vaccines that were given as mass vaccinations in response to these particular epidemics. The problem, as you can imagine, is that these responses oftentimes were too late. In other words, by the time the vaccine had been commissioned, delivered to the country, and the country distributing the vaccine to the areas that the epidemics were occurring, that just too much time elapsed. And that, frankly, the public health good that could come from vaccines was just not realized at all. The last point is that as vaccine manufacturers had developed new products with better meningococcal vaccines, none was being developed to address the biggest problem in Sub-Saharan Africa, that is Group A disease. Group A disease in the world was virtually unique to Sub-Saharan Africa, and there was no vaccine for it. As a result of this, the Gates Foundation provided funds and asked that this problem be addressed, frankly, more comprehensively than had been in the past. The project really began as a partnership between the World Health Organization and an NGO called PATH that has its home in Seattle, Washington. And WHO had been asked by ministers of health in Sub-Saharan Africa really to do something in terms of this catastrophe that was occurring certainly every year in areas that were absolutely unpredictable, and in these massive epidemics that occurred about every 10 years. International meetings were hosted by WHO, and a recommendation that came out of those meetings was that new conjugate meningococcal vaccines, that is, higher quality meningococcal vaccines, should be developed. And in June of 2001, the Meningitis Vaccine Project was created with Gates Foundation support as a 10-year partnership between WHO and PATH. And the goal of the partnership was to eliminate epidemic meningitis through the development, licensure, and introduction of a new conjugate vaccine specifically made for Sub-Saharan Africa. The project began, and I remember the first thing when I met at WHO in Geneva was that I felt uncomfortable that I didn't really understand the problem, frankly, from the African perspective. And so the first piece of work was done along with WHO headquarters and also the regional office in Africa, with consultations with African public health officials in Niger, Burkina Faso, Nigeria, and also at the regional office, which at that time had been in Harare. And everyone pretty much said the same thing. They said that meningitis was a very important public health problem, and that the biggest problem facing Africans at that time was frankly the cost of new vaccines. And when we explored the question of cost with ministries of health and also ministries of health, they made the observation that for this to be a successful project, the vaccine that was developed probably should not cost more than 50 US cents. That is $0.50 a dose. And this was at a time when equivalent conjugate vaccines, at least in the United States and in Europe, were costing somewhere in the range between $80 and $120 a dose. And that then, right from the very beginning, posed a problem. We began the project with the aspiration that we would be able to develop a partnership with major vaccine manufacturers that already were making conjugate vaccines. And so we began discussions with them. It was clear after several months that the price point was a major sticking issue. And the issue became relevant to the point that the major pharmaceutical companies could not and would not consider a price point at less than $1 a dose for a new vaccine. And it was at that time that the project, because it had stable funding from the Gates Foundation, elected to do something totally different. We decided to become our own company. And we began, that is, the Meningitis Vaccine Project, to assume the responsibilities of a vaccine manufacturer by manufacturing our own vaccine. There were two issues that really impelled us to move in this direction. One were the inputs from African public health officials on the importance of vaccine price, and also that WHO had separately commissioned a study that suggested that if you were making a lot of the vaccine annually, the price point for a new conjugate vaccine could be as low as 20 cents a dose. This meant that there was room in this particular area if we were to take this task on ourselves. This we did. And we immediately faced a major problem. And the major problem was the intellectual property that governed how these vaccines were manufactured. Conjugate vaccines require a carrier protein and the polysaccharide that surrounds the group A Neisseria meningitidis. And you have to work out a way so that you can combine the carrier protein with the polysaccharide, because that is what creates the new vaccine. And this new vaccine empowers an immune response that is much better than using the sugar alone. As you can imagine, most of the intellectual property for doing this particular work resided in the private sector. But we were contacted in early 2003 by researchers at the United States Food and Drug Administration that they had developed a new technique to conjugate group A meningococci, and that they were willing to transfer this conjugation technique to the meningitis vaccine project. And they were willing to do it virtually at no cost. And so with help from the FDA and the NIH, agreements were quickly finalized, and a technology transfer of the conjugation method was done to the Serum Institute of India, which in the interim we had chosen as the vaccine manufacturer that would produce this new vaccine. And this is what the business model sort of looked like. It functioned as a stable triangle. We had a firm in the Netherlands that produced vaccine-grade polysaccharide that we needed to make the vaccine. We had tetanus toxoid as the carrier protein that was already manufactured at the Serum Institute of India. And we now had a conjugation method that had been developed at the FDA and now transferred to the Serum Institute of India. This entire package was governed by a series of contracts that insisted that the target price for any transfer of this vaccine was set at less than 50 cents a dose. This is the partnership that led to the creation of the vaccine that went into field trials. It took 2 years to develop that. And the field trials began in 2005 and extended until 2010. There were 6 field studies. Those field studies were done in Senegal, Gambia, Ghana, and Burkina Faso, as well as India. The result of these studies was that the Serum Institute of India was granted an export license for this vaccine, now called Menafri-Vac. And the entire dossier was submitted to WHO, and the WHO pre-qualification team granted authorization of this vaccine in June of 2010. And later that same year, in December, the vaccine was introduced in Burkina Faso, Mali, and Niger, where 20 million doses were administered, and the transfer price for this vaccine was 40 cents a dose. This was our target area for this particular vaccine, and the vaccine that was unrolled in 2010 did so really quite successfully, but it offered the larger challenge was how do you expand this so that this new vaccine could be used elsewhere? Well, we had some help, and the help that we had were the clinical results for the introduction of this vaccine in Burkina Faso. And I'll spend a moment on this particular slide because it's important. These are the meningitis cases that were reported in Burkina Faso before and after the introduction of Menafri-Vac, or this MENA vaccine. You can see that in the years 2006, 7, and 8, there was a very large epidemic of about 45,000 cases of meningitis. And when you looked at the cerebrospinal fluid isolates from that particular epidemic, almost 90% of these strains were group A Neisseria meningitidis. So this was a very large epidemic of group A disease. And in 2010, about 25% of all positive spinal taps still yielded this particular organism. We vaccinated And look what happened. Cases fell dramatically, and there were no more Group A isolates that were identified in 2011, and that trend has continued to this date, with 2 imported cases that occurred in 2015. In other words, the vaccine was highly successful. As a result, countries really began jostling in terms of when can we get this vaccine as soon as possible. With leadership from the World Health Organization, a 6-year plan was developed that allowed the introduction of the vaccine to continue to unfold. And in point of fact, that entire scenario wasn't completed until, I would say 2021 or 22. But to make a long story short, 24 countries conducted mass vaccination campaigns of 1 to 29-year-olds. There were 350 million Africans that were immunized during the course of this vaccine release phase. And the most important result was that there were no confirmed cases of Group A in the entire meningitis belt. That is 500 million people. The last 2 cases were reported in 2018. There have been no cases since then. This major public health success has resulted in a lot of reflection, not only on my part, but on individuals that participated in this entire project. And what I wanted to reflect with you all are some of the lessons that were learned when we reflected on why and how this unfolded so successfully. And I'll go through these, some in more detail than others. I think most importantly, the first lesson is getting the science right. There is nothing more important in terms of the development of a vaccine to make absolutely sure that the vaccine is a quality vaccine and is an effective vaccine. We were able, thanks to the partnerships that the project developed, to access some of the best epidemiologic and scientific data in the world that could be applied to this particular project. This wasn't about making money. It was about making a product and eliminating a disastrous disease. We were lucky in that we had a model that we could follow, and that model was the introduction of the Men-C conjugate vaccine in the UK. And we had just terrific access to, again, the epidemiologic expertise that was available there. In short, all of our preclinical and clinical studies were consistent that the vaccine that had been developed was potent, safe, and was much better than polysaccharide vaccine. This is shown really in this slide. This is what's called technically a reverse cumulative distribution curve. And this basically measures the amount of protective antibody that's induced after inoculation with polysaccharide vaccine in red and the new conjugate vaccine in blue. Unfortunately, this skipped. I am not capable of going backwards. I apologize for that. In short, the vaccine behaved exactly the way it was supposed to, and that follow-on carriage studies showed a second component and a second, I should say, advantage. And that second advantage was that during the course of immunizing the age group 1 to 29 years of age, we were successful in eliminating carriage of this particular organism so that the protection in the population extended to everybody because the organism simply disappeared from the population. So that the science, again, were really the driving elements to make sure that we were moving in the right direction. Lesson 2 was the importance of African engagement. We observed from the very beginning, the African meningitis program is an African problem, and it's going to be solved by Africans. A new vaccine can come, but new vaccines don't do anything unless they are properly distributed and properly utilized in a public health sense. And here we were very successful. And the success was making sure that, number one, the vaccine was affordable as defined by Africans. And secondly, it was by listening and partnering with Africans from villagers to ministers. We were fortunate enough to have the president of Burkina Faso serve as the patron. And that did not occur through accident. We interacted with the president and we sought his help, and he generously provided his help. We established a field office, a large field office in Ouagadougou, and spent really a lot of time networking throughout the meningitis belt. And we invested in African communication expertise. Africans know how to communicate with Africans. And the investments that we made there really paid huge dividends in facilitating the introduction of this particular vaccine. Here is the president. This is Blaise Compaoré. And this was the day that the vaccine was introduced into Africa on December 6th in 2010 in a ceremony in Ouagadougou. The third lesson that I want to emphasize is the role of the World Health Organization. The project was a partnership between WHO and PATH, but WHO, particularly in terms of the epidemiologic studies and the introduction studies, played an absolutely critical role in the rollout of the vaccine in Africa. WHO is the most important immunization group in Africa. They were involved in every single phase of the work of the meningitis vaccine project. We had regular visits, at least 3 or 4 times a year, to African regional offices. We established an advisory group that was exclusively African, that served as an advisory group. It was led by Francis Nkrumah from Ghana and provided us with very useful information. Plus, the members served as ambassadors as we approached individual countries. The work with WHO required patience, tact, but it was an absolutely key element in MVP's success. The next lesson is partnerships. In doing something like this, this is just not MVP that accomplishes this. Partnerships are absolutely essential because of the scope of the problem that you end up having. And we learned important lessons as we developed partnerships. Number one, they have to understand individuals' partners' needs. Some needed money, some wanted recognition, some wanted public service. It really was different for each partner. But it was important that partners accept the general premise, that is, the premise that this was a public health, an urgent public health matter that had to be resolved within specific price points. The project I preferred, and the project preferred, face-to-face meetings in terms of binding partners together. And those meetings followed the same protocol. It was how did the partners see the partnership, and how were their contributions making a difference? Partners' contributions were accepted, and what wasn't working was probably the most important issue to be discussed so that these issues could be resolved and the partnerships made stronger. This is a pretty complete listing of all of the partners that we had. And if you look, I mean, it's National Institutes of Health, Médecins Sans Frontières. These were small groups like Axio or AMP. It included CDC. I mean, there were really a lot of these particular partnerships, and all of it was focused on the health authorities in the 26 countries in Sub-Saharan Africa and India. These formed really the floor for the partnerships to succeed or not succeed, as the case may be. What was seen by some of the partners was really some interesting opportunities. Serum Institute of India viewed the partnership with MVP as an access to conjugate vaccine technology and know-how, as well as improving their collaborations with WHO, UNICEF, and Gavi, as well as providing a product that really has a high visibility in Africa. And it lowered the financial risk that Serum Institute of India would have to take. When all the details were worked out with the project, the Serum Institute invested about $8 million, but again, the big hunk was the $37 million for the clinical trial costs in Africa and India, and those were completely shouldered by the Meningitis Vaccine project. And in the end, Serum Institute of India, because of the success of this vaccine, had international recognition. So from a partner standpoint, this is one of the partnerships that worked very well, in the same way that it worked very well for Gavi. Gavi was interested in supporting successful vaccines that are affordable, and the Menafrivac vaccine fit perfectly within their price point. And at the time that the vaccine was introduced, we didn't exactly know how effective it was. But once we had the data from the first couple of years, then, and also the fact that African countries wanted Menafrivac, the partnership with Gavi literally flourished. Those are the, I would say, most important lessons. We had some other observations, and I would pass these on to you. One observation was, when something is not working, do not rely on the internet. Go to the field. That's where the problems are, and that's where problems are resolved. And it turns out you just save an enormous amount of time by actually going to where the problem is. The other thing is that we had consultations with experts, and it became clear after a while that experts didn't know everything. And so it was sort of somewhat refreshing to have individuals suggest something that you didn't follow and then succeed. The other is the observation that partnerships that come into it, to quote unquote do good, that's not enough. And we quickly learned that the economic models have got to make sense to all partners. That doing good is good, but it doesn't carry you all the way. The other thing is that we soon learned that as with international products that are being developed, regulatory processes are extremely complex, and it's never too early to begin work with regulatory officials. Lastly, in any complicated project that's international, it's safe to expect surprises. We had plenty of surprises. And working through surprises oftentimes is very illuminating. And we learned that one of the solutions is to make absolutely sure that our lines of communication were always open to all of our partners, so that if surprises were going to occur, we got the surprises earlier rather than later. Lastly, what was our report card? What were the costs and the impact? And here we have pretty good data. The total vaccine development cost for this brand new vaccine worked out to be about $100 million. And that $100 million was furnished 100% by the Gates Foundation. The vaccine was widely accepted, and over 350 billion Africans have received a dose of Benafrivac. And that as of December of 2022, the vaccine has now prevented as many as 1 million cases of bacterial meningitis in Sub-Saharan Africa. And the disease, that is Group A, Neisseria meningitidis disease, has disappeared. And lastly, we've had economists that have actually worked through their magic models and have shown us that the vaccine has saved money. Up until the end of 2022, it's estimated that the vaccine has saved about $500 million to date, and that will continue, again, with the continued absence of Med-A disease in Sub-Saharan Africa. Lastly, one of the things that we were very worried about was making sure that we created a vaccine that was used right away. There were two examples in global public health. One is hepatitis B, the other is the Hib vaccine, the meningitis vaccine for the under 2 years of age. These were vaccines that were available in the United States in 1982 for hepatitis B, but weren't used in developing countries until 2001. That was a gap of 19 years. It took 19 years to use this product in areas that had much higher disease rates than in the United States. Same story with hepatitis B. That took 11 and 7. It took 18 years for it to be used at public health scale. With the Men-A vaccine, it was used at public health scale the year the vaccine was licensed. There was absolutely no delay. in getting this vaccine to where it was needed. This is my last slide, or second of the last slide. This is the team that developed this met only once. And this was as a result of a generous grant from the Merieux Foundation that helped pay the travel costs. And we all met in India just before the vaccine was introduced, where we reviewed where we were just prior to the introduction of the vaccine. This is just a wonderful, wonderful group of people that work very hard and work well together. And I close with the customer. This was the customer for this work, and will always remain to me, or at least for me, an inspiration for why we do public health. That pretty much finishes what I have to say, and I am going to stop share and
Thank you, Professor Laforce. You don't need to mute yourself, Mark. I think we need you to answer the questions.
Okay.
I have, thank you very much. I think that's very enlightening, and just to see what's the importance of public health concepts to be applied on such important projects. I have many questions, so I will try to combine a couple or two questions together, or three, so make it faster. Now, one of the important aspects that the participant want to hear about, is it hard to duplicate such project with another infectious disease?
I would say that's a very interesting question. And again, the project was really quite lucky because it was focused on one agent that was causing an enormous amount of disease. And so finding a solution to that one agent is what I just described.
Right.
If you now say, well, okay, can you translate this to diarrheal disease, for example? In other words, the syndrome of diarrheal disease. Yeah. Well, the answer is no, you can't, because it is much more complicated, because you have Shigella, you have rotavirus, you have cholera, you have so many agents together and that's why when I said at the beginning, make sure you get the science right. It means that you have to have an in-depth understanding of the causative agents for what you are trying to accomplish. And unless you have that information well in hand, then it is very hard to design an answer. And it is for that reason that I think the scientific base for moving in a particular direction is absolutely essential. Okay?
Thank you. Yeah, I think that's probably the expected answer. So it is one organism causing a broad area involved with such disease, while other disease, as you described, can be caused with many other agents, viruses and bacteria.
Correct.
Now, what's the other source of funding other than philanthropy? Okay, is there any other possible source to get the fund?
That good question. The answer is yes. And the main source of funding, has been really a large chunk through philanthropy, with the Gates Foundation being certainly the new customer. but there is no reason whatsoever why gates should exist as frankly, there are potentials for any philanthropic group that has access to funds. What I happen to see is that oftentimes those funds end up being frankly misdirected because of the absence of a scientific base for the correct utilization of those funds. That's item 2. Item 3 are governments. Governments have usually very large resources. The problems with the resources in governments is the politics. And frequently the politicians who really aren't scientists don't defer to scientists, and you end up with all kinds of projects that oftentimes are more wasteful than useful. And so it really, this is why I always feel really impelled in terms of saying this. If there was more time that were spent by governments in terms of trying to establish proper areas to go, in other words, where the science and where the epidemiology can take you, I think that you would end up with, frankly, a better utilization of the financial resources that are available. I know I didn't answer the question and I sort of ran around, but that's the way it is right now.
That's the reasonable answer. I have many questions here, 3 questions back to back, all from colleague from UAE University. So let me select the easy one first. The question is, in the UK, when they had a vaccine available for group A and C, then more cases of group B emerged. In your experience in Africa, is there any increase of other group strains while group A has been eliminated?
Good question. And that was something that we were very worried about. And the answer is that the introduction of the vaccine and the elimination of A has created no special niche for other meninge. We know that the other strains that are circulating, group X, group C, and group W, continue, but there has been absolutely no change in their epidemiologic patterns with the absence of group A. Secondly, when we were doing the carriage studies, we were able to show that the level of carriage was really quite low, despite the fact that we saw a lot of disease. So it meant that this small little area that was only about 1%, even if that were filled with another agent, is not going to create anywhere near the level of disease that was created with Men-A.
No.
So those are 2, I think, quite specific answers to that question.
What an advice to convince people in some of the African countries and Asian countries to take the vaccine? There are any specific advice?
Okay, the specific advice in Africa, this vaccine was extremely very well received. There was the coverage rates done by independent individuals going into the country after the vaccinations had been done in 1 to 29-year-olds showed on average that the vaccination rates ranged from 92 to 96%. That means the vaccine was essentially accepted by everyone. We had absolutely no problems in getting the vaccine utilized by people. You might say, well, why? Number one, Africans hate meningitis. Meningitis has ruined so many families that they knew this as a major event that tended to happen in individuals between the ages of 8 years old and 18 years old. These are the age groups that are incredibly important to families because these are young men and young women that are about ready to begin their adult lives. And so resolving that problem was really a major societal thing. The second issue was that we invested, as I said before, in communication groups in Africa. And they developed a way that I thought was quite clever. The French word for good is bon. And the vaccine was as families spoke to each other, this was the good vaccine, le bon vaccin. And so this was sort of like a catch-all word that, oh yes, you were getting le bon vaccin, the good vaccine, not the other vaccine. This was the good vaccine. And so that's what mothers basically, when they talk to each other, because it's mothers that make the big difference. That was the other thing that we learned, is that frankly, within the African culture, if you could convince the mothers, essentially you were home free. Now, it may be different elsewhere, but those were the two approaches that really minimized individuals not wanting the vaccine.
Thanks. I have one long question. Probably I will divide it to two, so it's easier for me to, or easier for you to answer. It's in fact a couple of research, one from Burkina Faso and one from Ghana. This is the question raised by Dr. Sheikh Hussein from your university. The first study from Burkina Faso reports that atmospheric dust does not associate with occurrence of meningitis epidemic, which might be another triggering factors. So, do you want to answer this, or shall I give you the other part? So the other study,
I know I'm happy to wrestle with that at first. This is really a very important question because. Again, let me take one step backwards. When we tried to understand why these epidemics occurred in Sub-Saharan Africa, there were a couple of observations that were very important. Number one, they always occurred at the same time each year. They were between weeks 2 and week, I would say, 16. And that is the driest time of the year. And it's the time of the Harmattan, where the winds come off the Sahara, and it turns out to be very, very dusty. And this has been reported since long time ago, that once you had the first rains that occurred in usually May, or certainly by June, but usually in either late April or May, epidemics that were ongoing immediately stopped. They immediately stopped. Mm. And so it meant that there is some relationship about either the dust or the relative humidity in terms of how Group A Neisseria meningitidis are spread from person to person. And there have been descriptive studies that have looked at the amount of dust. There have been descriptive studies that have looked at relative humidity, et cetera, et cetera, et cetera. And there have been no definitive answers that have been provided to explain this particular phenomenon. And so, and it is one of the failures that I would say of the project. I spent a lot of time trying to convince these very sophisticated researchers at the Pasteur Institute in Paris or at NIH or at Cambridge or at Oxford. What I wanted them to do is to send complete teams of scientists to sit through one whole epidemic season and to do their magical studies to try to explain why the epidemics always occurred at that moment in time. I failed 100%. I never convinced anyone to make that particular special trip. And so I'm left, and I realize that I'm probably going on, but as you could tell, it's something that really has bothered me because there is still no specific pathophysiologic explanation for why this occurred. The only thing that we know is that the vaccine has solved it. That's it.
So probably you already answered the second part. So the other research part, in fact, is a study from Ghana showed a significant association between climate change and meningitis outbreak. So that's the question is always there, where we need to draw the line. Okay, so is it a factor or not? So that's many times, many things in science probably there, you don't have a real answer to it. That is correct.
That is correct. And I think we have enough scientists. It's just that we have not been able to find, pardon me, the interest and the money to actually get that done.
Many questions I have to choose. We left with the 3 minutes, so probably I can choose one more question here. I will not go. There are many about the application of the similar, so I will not ask about they give me another example. For example, HPV. Can we use it as another possible vaccine, preventable disease? But there is another one. Another question is about: do you believe that the healthcare official learned from this project upon implementing it in recent years?
Oh, I think the answer to that is yes, because the meningitis epidemics were one of the reasons that ministers of health lost their jobs. And so, for one thing, ministers of health really feel much, much safer nowadays that the vaccine has eliminated these large epidemics. So my answer is that yes, there has been a general, frankly, sigh of relief on the part of individuals that have had to face these particular epidemics. But from my standpoint, I find the sense of relief when I've talked to mothers and when I've talked to fathers and when I've talked to just Africans in just over a cup of tea or just chatting with them. They basically say, thank you.
One more last question, probably this is the last. What was the role of our microbiome signatures? I heard about the microbiome a lot during the World Government Summit that it just concluded today. And there are many interesting talks about the use of microbiomes in treatments. So do you think there are any role?
I think when it comes to meningitis and the information that we've obtained in terms of the group A difficulty, I don't think the microbiome has anything to do with this at all.
Thank you very much. And I think that there are many other questions, but probably the time to conclude the session. Thank you, Professor Marc, for this very important topics to be discussed and give an example actually for future projects, something similar to that. Thank you very much.
Thank you.
Thank you, colleague, and please I would like to thank Professor Laforce for this interesting topics. I will remind you again that all attendees will receive an email to download the attendance certificate within the coming days. The next webinar, inshallah, of the series will be hosting Professor David Hu is the winner of the Hamdan Award for Medical Research Excellence for Immunity, who will address his work in the HIV research. So there are a couple of HIV questions today, but intentionally I decide not to go through these details, and I will leave it probably for the next webinar. Thank you very much for your kind attention. Thanks.